Hello everyone, I'm Koyan and today we are going to learn how to answer questions related to Form 4 Chapter 3 Movement of Substance across Plasma Membrane. So before I enter this this question, I would like to show you the recap of the movement of substance because there are many types of substance and the way they enter the plasma membrane all different pattern punya. So let's take a look. There are lipid soluble, let's say the fatty acid for example. Yeah, it actually moves through the phospholipid bilayer straight away without the help of channel protein or carrier protein. So when it's straight away diffused inside is called simple diffusion. And for non-polar small molecule like carbon dioxide, yeah, this one it also enter the plasma membrane through the simple diffusion. You just move across the phospholipid bilayer. Meanwhile, same goes to water, just that water, it needs this process called osmosis. It's different name, but the way they enter is also the same. It's also a passive transport, but yeah, you have to call it osmosis. And for large molecule, yeah, since they are too large to pass through, they have to use facilitated diffusion. So they need this carrier protein help. So carrier protein, they basically will look like there is like a fake shape like that one. Yeah. Meanwhile, for channel protein, they normally have like this is the channel protein. They just really let substance pass through. So large molecule they require carrier protein like the glucose. Meanwhile, for ions, they require either one of them, channel protein or carrier. It depends on the situation. So for channel protein, yeah, they just go through facilitated diffusion. Meanwhile, for carrier protein, they require active transport. And you have to know what are the differences between passive transport and active transport. The most obvious difference is that active transport, it require energy. Meanwhile, for passive transport, it does not require energy. And then this one is for passive transport. It moves down the concentration gradient, means it moves from a high concentration to low concentration. Passive transport. Meanwhile, for active transport, yeah, it moves from a high to a low concentration. That's completely the opposite way. So let's take a look at this question that came out in Terengganu paper. Diagram shows structure of plasma membrane. State the name of parts labeled P and Q. So what is actually this small thingy that float on top? It's not float like it's actually attached to it a bit. So this one, this is actually a protein, but we didn't learn in detail. It is actually a type of protein where we call it as extrinsic protein, but you don't have to remember about this because you only learn in further detail if you continue to study biology in college or university. Meanwhile, for this one is the intrinsic protein. So for SPM syllabus, right, we only focus on the channel protein and carrier protein, which is the intrinsic protein. But no matter what, you don't have to panic because this one and this one, they are the same protein. I mean, not same protein. They are also made out of protein. So since this thingy is attached to the protein, so it is this carbohydrate chain. This is actually a carbohydrate hydrate chain. So it attached to this protein. So it is called as glycoprotein. Glycoprotein because it's attached to this protein. Same goes to this. This is also a glycoprotein because it's attached to a protein. Some students don't know how to differentiate between glycoprotein and glycolipid. So what about if there's a carbohydrate chain that attach to the hydrophilic head? So this is a hydrophilic head. So it attached to it. So this one, if it attached to a hydrophilic head, or you can say as polar head, this one is actually the glycolipid. Yeah, that's the differences between them. You have to see which one it attaching to. So if it attached to chain protein, all this, they are actually glycoprotein. If it attached to a polar head, so it's actually a glycoprotein. And moving on, what is actually Q? Q is so a lot of students get confused. Phospholipid polar head hydrophobic tail all these things, right? So let me clarify with you. How do you actually see this whole thing? Okay. All right. So if they label this thing, they want you to, they ask you what is this? This thing, the head and the tail is a phospholipid. Okay. But if they label this pair, not pair lah, but two layers of it, it is actually a phospholipid. You have to add on something. Phospholipid bilayer because it has two layers. So it is phospholipid bilayer. You must write the bilayer out. Means it contains a two layer of phospholipid. This one is just one layer. So it's, you can say as phospholipid layer lah. But if they just ask this one of them is a phospholipid, meanwhile, if they just point out this, so this you can answer it as a polar head, or you can write it as hydrophilic head. If they point out the tail, it is a hydrophobic tail, or it is a non-polar tail. Alright, so let's move on to the next question. P is formed from combination of carbohydrate chain that attach to protein molecule. So state the function of P. What is actually the function of glycoprotein? I know a lot of students didn't really remember much about the function. So for glycoprotein and glycolipid, they actually normally have the same function from what we learned in SPM. Just that the how they attach to is the thing that attach to is different, but their function is the same. So for this glycoprotein, their function is actually to, there are three functions we actually learn lah. So it acts as a receptor molecule for hormone. Another one is to stabilize the membrane by forming hydrogen bond in detail lah, by forming hydrogen bond with the water. It will actually stabilize it, bond with water. This is a more further explanation, but most importantly is to stabilize the membrane. And then it also acts as antigen to cell identification. So this one is a very detailed picture. But if we actually look further, right? Okay, let's say this is the animal cell. So this animal cell, they actually have this thing called a receptor. So you know how the hormones actually transported and go to the animal cell. So let's say one of the hormone. So hormone, they actually bind to the receptor. So this is actually what your glycoprotein do. It acts as a receptor. And then your hormone is this one. So it binds to the receptor. And then, yeah, it will initiate a cell signaling process for your cell to do a specific action. But we didn't learn that in detail. I'm just letting you know what is the meaning of as a receptor molecule for hormone. Another reason for the as act as antigen to cell identification It's basically like when they want to identify the cell, they actually identify through the antigen present on the membrane. Yeah. So let's say another cell wanted to identify this cell, they will actually identify through the antigen that present there. So this glycoprotein, it can act as an antigen for the cell identification. So maybe it had this specific structure that, oh, they know this is what type of it. So how to identify the type of cell? So it's act as an antigen of it. Now let's continue. Plasma membrane has semi-permeable membrane characteristic because it is built from layer Q. So layer Q is the phospholipid bilayer, and protein state 1 characteristic of substance that able to move across layer Q. So it can be a small molecule like the carbon dioxide, or it can be the lipid soluble. So yeah, small molecule. Or you can write it as lipid soluble. Now let's move on to C. Saline solution is a solution used to treat diarrhea patient. Explain why. So for diarrhea patient, they actually lose a lot of water. So in order to illustrate this, let's take a look at this large intestine, although I'm not really good at drawing. Okay, so let's say this is the large intestine. So let's take a look into more detail lah. So in this large intestine, what is the function? They are actually want to do reabsorption of water, right? Before it become a feces, right? So they will do reabsorption of water. And yeah, if you reabsorb too much water, right? Or maybe your feces stay in your large intestine for too long, they will keep continue to reabsorb water. So the water from the stool will become reabsorbed to the blood vessel. So your stool will eventually become very hard. Okay, it's not soft. But what about diarrhea patient? So for diarrhea patient, they actually have bacterial infection in their large intestine lah. So it's invaded by the bad bacteria. So for this type of situation, their water tend to lose out because here got a lot of let's say got a lot of chloride ions, got sodium ions. So here got a high solute potential, means it has a low water potential. So instead of reabsorption, most of the water will eventually move from a high water potential to low water potential. So the water will actually move out from the epithelial cell. And then guess what happened to your feces? It will become extremely watery because all the water you lose out from the epithelial to the large intestine here. So yeah, it leads to a build up of water. So when water mixed with the feces is extremely watery. So yeah, and other than that, because the reason why we do reabsorption water is to prevent dehydration, you need water for a lot of metabolic process. But right now you are actually losing a lot of water. And a lot of some of your ions right there in your large intestine, it will all be channeled out through your anus. So you will eventually lose a lot of water and electrolytes. So the reason why they need a saline solution is to replace the water that is lost. Replace water and electrolyte that is lost. But for saline solution, right, it's actually for diarrhea patient that is very severe. They have to admitted to hospital and then get the IV drip. So the IV drip consists of this saline solution. And other than that, why do they use saline solution instead of salt solution or normal water? Because saline solution got a specific content of the solute and water lah. They can't be like 100% water or very high concentration of salt. It's because our red blood cell, you have to maintain your red blood cell in an ideal condition, or else it will get cremated, it will get shrink, or maybe it will get hemolysis, it will burst, your red blood cell will burst. So saline solution, they are actually kept in a ideal situation where your red blood cell is in the isotonic solution. So it won't burst, it just retain in its shape. So yeah, that's the reason why you have to use saline solution. So in biology way to describe this saline solution is that it is an isotonic solution to blood plasma. Alright. And then it can replace water, or you can say recover will be better lah. Okay, recover water and electrolyte loss from patient. So the patient won't be dehydrated. So it undergoes rehydration. Rehydration, not dehydration. Because for diarrhea patient, they lose a lot of water when they excrete their feces, because the feces is extremely watery and contain a lot of solute. So yeah, they will lose a lot of water. In order to recover water, they will use this IV drip saline solution to recover the loss. So they will like put it in an IV drip lah. But for a mild condition of diarrhea people lah, normally we would just drink the oral rehydration salt. So later on the question we mentioned about it, then yeah, I will explain to you. Now let's continue the next one. So two types of transport in living organism. So it's actually related to chapter 9. Yeah. And this one is talking about the one in your kidney. Okay, now let's take a look at this fructose. So when the fructose being absorbed into your blood capillary from your small intestine, then absorbed by the villus. This one is actually through a facilitated diffusion from a high to a low. Okay. Meanwhile, for this one is the this is actually the glomerulus, and then during this situation is where ultrafiltration occur, and then reabsorption occur. And during this process, the glucose is 100% absorbed into the blood capillary. So 100% means they are completely absorbed. So this one is actually from this one is actually using an active transport. So eventually you keep absorbing no matter what. Yeah, it lead to, because they want to maximize the absorption, so yeah, they will use this active transport to absorb it. And so this one is basically facilitated diffusion and active transport. So this one requires your knowledge from the chapter 9, and this one is about the formation of urine, all those thingy, those processes. Now let's take a look at this part. Differences between two types of transport named in 1AI. So what are the differences between them? So facilitated diffusion is actually a passive transport. So it occurs down the concentration gradient, means high to low. So occurs down the concentration gradient. Meanwhile, for the active transport of glucose, it is occurs against the concentration gradient. Since this one is active transport, it requires energy. This one is very direct, and this one does not require energy. And when it keep occurring down the concentration gradient, it will occur until a dynamic equilibrium has achieved. So another point I can add on for you for more option, until a dynamic equilibrium is achieved. Meanwhile, for the next one is there will be accumulation and disposal of molecule. It will lead to an accumulation basically, or maybe a disposal or ions. Alright, so normally how teacher mark is they will mark through, if these two are correct, then yeah, this will be given 1 mark and this will be given 1 mark. But you don't have to draw another box lah. You only give up 2 of your most confident answer. Now let's move on to this one. Diagram 1.2 shows experiment to study effect of oxygen in the soil water through the transport of mineral by root hair cell. So explain why mineral salt is less than, for the A is less than that B after 7 days of experiment. So when you want to study the effect of oxygen, you can see soil water with the presence of oxygen, and this one doesn't have oxygen. So in the end, the mineral salts in diagram 1.2a, they get to be lesser. I mean the soil there, it has a lesser mineral salts. This one, it got a more mineral salts. The soil solution that without oxygen, it still has more mineral salts. So what can you give in conclusion? Alright, so you have to actually know what happens in the root hair cell right there, the root cell. So for the root hair cell, right, okay, lama. Okay. So let's say this is one of the root hair cell, right? It got a lot more cell. So I just draw one of it. So for root hair cell, they, in order to maximize the absorption, right, they will actually they will need oxygen to actually generate energy through cellular respiration. So why do they need this energy? So when there's the presence of oxygen, they can carry out cellular respiration. So cellular respiration, you get to generate energy. So why do you need this energy? Because this energy is required for the active transport of mineral salts. So mineral salts, let's say, they will get actively transport into the root hair cell. So they all get actively transport into the root hair cell. So once it keep continue to get actively transport to the root hair cell, now it has a high solute potential because it has a lot of mineral ions right here. So leading to a low water potential. Meanwhile, what about the water on the out at the soil solution right there? So the water outside, it will be a high water potential. So the high water potential, it will, yeah, diffuse into. So the water molecule will diffuse into the root hair cell via osmosis. So basically, when you inhibit, when you didn't provide oxygen to the root, they are unable to carry out cellular respiration. When they are unable to carry out cellular respiration, they can't provide energy for the active transport of the mineral ions. When there is not much active transport of mineral ions, you know that there is still many mineral salts in the soil solution right there, containing still remain at there. Meanwhile, for the others, if you provide oxygen, there will be more energy produced and then more energy for the active uptake of mineral salts, mineral ions. So they will keep absorbing the mineral ions. That's why when you keep absorbing, of course, your soil's water will have a lesser mineral source. So how do you answer this question after such long explanation? Is that mineral mineral salts are actively transported into root hair cell. So presence of oxygen in soil, presence of oxygen in soil water allows the root hair cell to undergo cellular respiration to generate energy. So yeah, this is how you explain it. So when you are explaining it, you focus more on the diagram 1.2a situation where the mineral salts get to be absorbed, which means actively transported to be precise. And then, yeah, why presence of oxygen will lead to it. Cellular respiration to generate energy. Now let's take a look at the next question right here. Shows two sucrose solutions with different concentrations that are separated with a selectively permeable membrane. So it has a 30% and the others has 10%. So the sucrose is the big molecule right here. Alright. Then they want you to identify what is the solvent and what is the solute. So solvent is the water. Solute is the one that dissolve in the solvent to form a solution. So let's say lah, okay, this one you got a water. So it is a solvent. Then you go and add on sugar inside. So sugar is a solute. Okay, so sugar plus sugar, which is your solute, plus your water solvent, it will form a what? A solution. So you call it as sugar solution. Yeah. So this is how you differentiate between them. Okay. And then draw to show the direction of movement of water molecule that will occur. So this one has a low solute concentration, right? Because 10%, this one got 30%. So here got high solute. And then for water, you can see here got a lot of black, yeah, the black square. So yeah, it has a higher water potential. Here got a lower water potential. So it's obviously water moves from high water potential to low water potential via osmosis. So yeah, you basically draw this arrow and then you get your mark. Okay. Since this one is a selectively permeable membrane, you have to know that sucrose are unable to pass through it. Only the water molecule are able to pass through it. So yeah, the number of sucrose still stays the same. So in order for the water to achieve a dynamic equilibrium, yeah, the water will actually move from a high water potential to lower water potential. Now let's take a look at this question. So it's a very long newspaper, but don't worry if you can't see what's inside the context. They don't expect you to read it one by one. You will be wasting your time. So it basically shows a newspaper talking about a group of researchers that successful in developing SILRH membrane distillation. Okay, so they are treating the seawater using a membrane. So it's a membrane made from paddy chaff and can be used for desalination process. Desalination means you remove out the salt from the salt water. Yeah, desalination. So name the technique used in desalination of seawater process. This is actually in the application part of this chapter. So yeah, you have to know this process. This technique is actually called reverse osmosis. RO, reverse osmosis. So what is the role of membrane in desalination process? So you want to remove the salt from the salt water. Of course, you need something to filter it out now. You need a membrane, no? Cannot just straight away like that, give you drink. No, you need a membrane to filter the salt out. So you have to mention what are the characteristics of the membrane and then what happened to it. I mean, what you do to the water so that, yeah, you get filtered out. So the characteristic of this membrane is that the membrane is a selectively permeable membrane. Permeable membrane. Selectively permeable means it can allow some substance to pass through, some substance are not allowed to pass through. And then, okay, pressure will be applied to push the seawater through the membrane. So let's see. This is the membrane. And then here got a lot of solute, a lot of solute. So you actually need to apply a pressure right here to push so that, okay, the salt won't be able to pass through, the solute won't able to pass through. So the one that can pass through is the water. So yeah, the water are the ones that able to pass through. Meanwhile, the salt still stays behind the membrane. Yeah, they can't pass through. So you have to use pressure. You have to apply the pressure to push the seawater. So this membrane, the membrane will only allow a specific molecule. I mean the water to pass through lah. Not specific molecule. It only allows the water molecule to pass through. To pass through, which means that it will prevent other particles foreign particles, foreign particles, salt, microorganism to pass through it. So in the end, what is the product you get to produce? you actually get to produce a pure water lah, pure fresh water. It's obtained, it's released, obtained. All right, let's take a look at the next question for this. Draw the level of both solution after dynamic equilibrium is achieved. So remember the sucrose unable to pass through. They only allow the water to pass through. So the sucrose, they still have the same amount. You must draw the same amount. You cannot add on extra 1 or extra 2. No, cannot. So this one got 3 and the next one 1, 2, 3, 4, 5. So you still got 5 of them. But for the water level sure change, right? So for the water level, since here got a lesser solute, means here got a higher water potential. So here got a lower water potential since it has a higher solute. So high water potential move to the low water potential via osmosis. So yeah, you know that the water level will increase for the right hand side, just like that. Okay. Now let's see this question about what happens to the red blood cell after you immerse into a type of solution, but they didn't let you know what type of solution. You have to predict based on the shape of the red blood cell. So does your red blood cell actually looks like this? No, right? Your red blood cell don't look like this. It actually is supposed to look like a biconcave shape, biconcave disc shape, not this weird shape, spiky shape. So you know that this red blood cell have undergone crenation, is crenated. Okay, if they draw you that the red blood cell it gonna burst already, like they got burst, they draw that the red blood cell is swell up and then it bursts. Yeah, this one is hemolysis. So hemolysis, this actually used for the red blood cell only since it only write hem. I mean it's hemolysis. Okay, plant cell it won't undergo hemolysis. Okay, because this is for the red blood cell. The term like more specific, that's why it's called hemolysis. Okay, this one is crenation. So you know what is wrong with the solution, right? So you have to write what happens to the red blood cell. It undergoes crenation. That's the first point you can get. So how do you want to explain? There's something wrong with the solution, right? So the biological way to explain this solution is that this solution is a hypertonic solution. Red blood cell immerse in a hypertonic solution. So the red blood cell, let's say lah, here is a hypertonic solution. So means that outside got a very high solute potential. Means it has a low water potential. So water in the red blood cell is a higher water potential. So high water potential will diffuse out. I mean the water will diffuse out of the red blood cell. That's why it will eventually shrink. So water diffuses out. Water molecule diffuses out from red blood cell via osmosis. The diffuse word and the osmosis word is really important for you to mention to get that point. Red blood cell shrink. Yeah. So here are the 4 points lah, but as long as you mention 2 of the points, yeah, you are correct already. I mean, you get full marks already. And last question right here is about oral rehydration salt. Just now that question is about saline solution, where normally you just like the nurse you insert it inside your uh vein there, and then the IV drip is actually containing saline solution. That one is more severe case lah. But if a mild diarrhea case, right, we normally just use drink oral rehydration salt prescribed by the doctor. So state the name of salt R and describe how salt R helps in reducing the effects of diarrhea. So it is a oral rehydration salt. That's the first mark you can actually obtain easily if you still remember the name. So what is the use of oral rehydration salt? Why can't we just use other thing? Okay, so oral rehydration salt, it actually encourages the, okay, most importantly, the point you have to write is to recover the loss of water and electrolytes. Since most of the water, wait, most of the water in the large intestine, it diffuse out. So your stool, your feces right now is extremely watery because the water diffuse out already. So yeah, you know you are experiencing dehydration. So you need to recover the water and the electrolyte loss. So dehydrate body to avoid dehydration. Okay, so I hope you can understand the difference between the saline solution to treat diarrhea patient and this oral rehydration salt lah. Okay, this one is just you just drink it lah. The saline solution is not for drinking one. That one is straight away insert inside your vein because saline solution is isotonic solution. So it will maintain your red blood cell shape lah. It's not hypertonic solution. Yeah, so your red blood cell won't undergo crenation, those type of worst case scenario. But yeah, both saline and the oral rehydration salt is actually to recover the loss of water and avoid dehydration is to rehydrate the body. Okay, so that's all for today's lesson and I'll see you in the next video. Thank you so much for watching and yeah, all the best. Okay, see you. Bye bye.